Trpevski? Trpevski? You have pronounced it correctly?
Yes.
That's a Polish name, right? Is it a Polish name?
No, but it's a Slavic name, that's true. It's,
okay. So, and he will talk about adenylyl cyclase 5 regulation enables detection coincident neuromodulatory signal in the striatum.
Yes.
Yeah, I didn't say
changed
that
the
very
title
nice.
a little bit. It means basically the same. Hopefully, it's easier to understand it. Can you hear? Yeah. So this talk for today is also a multi-scale approach to brain research in neuroscience. So it's a kind of an example of what Jean-Pierre and Joe was talking yesterday, and also maybe Antoine. And it involves adenylyl cyclase 5, which is an enzyme in the basal ganglia of the brain, which is involved in learning. And I work in the lab of Jeanette Hilger and Trpevski. This is in Stockholm. It's a computational lab. It builds models on several different spatial and temporal scales of the basal ganglia. And we are going to look at the intracellular signaling cascades now, so that level, and also go down to even molecular dynamics. Oh, yes. Thank you. Yeah, so the colored regions of this image are the basal ganglia, everything except this green part here, which is the thalamus, and the yellow part. So these are one of their roles in the brain is reward learning, and reward learning is the learning of new behaviors which lead to some kind of a reward from the environment, which is usually beneficial for survival and reproduction, and this can be food or water or pleasure coming from sexual intercourse or parenting, or so things like this. And the reward learning is the interest of the lab. And when it comes to learning, synapses are the prime, or one of the prime candidates to implement learning, at least the way the neuroscientists see it. And when it comes to reward learning, the synapses really engaged are the synapses between the cortex and the striatum, which is the input of the basal ganglia. So the interpretation that I'm going to talk about here is the way things are interpreted in neuroscience. So these are the medium spiny neurons, these spiny cells here, which have the synapses coming in from cortex, and also, so they receive cortical inputs and also neuromodulatory inputs such as dopamine. And those are the ones that we are going to work with today, or focus on today. Here is a schematic of such a synapse, so there's many molecules in a synapse, and they do many kinds of different things, but since we are interested in reward learning, we focus on the molecular circuitry that determines whether to strengthen or weaken a synapse based on this input. And in this project, we focused on strengthening, determining when a synapse is strengthened. And there's, if you simplify this, there's kind of two modules of this molecular circuitry here. So this one responds to the cortical input. Cortical input usually translates to calcium influx inside the cell, and if this calcium is strong enough, it causes the insertion of receptors in the synaptic membrane, thus strengthening the synapse. However, this effect of calcium only goes through if there is enough cyclic AMP produced in the cell. So cyclic AMP is critical for enabling strengthening of the synapses, and it is produced, its production is controlled by this module, and this is what we studied in this project. So neuromodulatory input, and this neuromodulation here is done by molecules, so not ultrasound or other mechanisms. This is what what we study in this project. Here is a zoom in of this module. Adenylyl cyclase 5 is the enzyme that produces cyclic AMP. It is regulated by two G proteins. This is Gαolf, which stimulates it, and GI, which inhibits it. And so this is the realm of G protein signaling. This is slower than membrane excitation, but faster than signaling that needs to go through the nucleus. And a brief recap about G protein signaling. So the ligands come in. These are the so-called first messengers. They come, they bind to receptors. Then the receptors can activate the G proteins, and the G proteins can exert their regulatory effects. Signaling is terminated by other GTPase proteins, which deactivate the G proteins. These are not shown here. And for the case of the striatum in the basal ganglia, so we here will focus only on this one type of medium spiny neurons, these D1 type of D1 MSNs. Dopamine is the neuromodulator which stimulates the Gαolf branch, thus can activate AC5, and acetylcholine is the one that stimulates the GI branch, thus can inhibit AC5. Both of these neuromodulators have a basal tone in the basal ganglia, and this is due. So this basal tone is due to the tonic activity of the cells which produce dopaminergic and cholinergic cells which produce these neuromodulators. And another thing characteristic for the striatum is that there's lots of these fast GTPases for GI. So we will assume in this project that GI is deactivated very fast. And then another thing about the neuromodulators is that there's also deflections from these tones in terms of peaks and pauses in these signals, and these have been seen to correspond to some quality of the cortical stimulus coming in from cortex, in the sense of a dopamine peak has been seen to represent a rewarding stimulus coming in. The cholinergic pauses here, it's not known what exactly they represent. In other regions, they have been associated, or cholinergic signaling has been associated with new stimuli, with novel stimuli. In the striatum, it has to do, one of the things is related to the timing of the stimulus, but it is still not resolved. And what is known from experiments is that in order to enable strengthening of the synapse, you really need dopamine here to activate this branch. And this is kind of normal. You need to produce this second messenger, cyclic MP. And what we were asking ourselves in this project was whether signaling through both branches somehow interacts at the level of AC5 to control cyclic MP production, and thus when the synapse can strengthen. And these receptors are indeed actually co-localized on the synaptic surface, and they even more, they form oligomeric complexes with the enzyme. So this kind of makes sense, and it was also a prediction, a possibility highlighted by earlier modeling studies of the lab. And this is what we went through in more detail, and I can tell you briefly the result was that indeed signaling through both signaling branches is necessary to activate cyclic MP production, and this is why. So it is known also that in the striatum, the basal tone of acetylcholine is enough to keep the GI branch active. So under basal state, resting or whatever, this branch is active. AC5 is inhibited. It is not enough to only have a dopamine peak. This will activate this branch. It turns out this can form, according to the results we got, this can form a ternary complex between the enzyme and the two regulator G proteins, and this one is inactive, cannot produce any cyclic MP. So you need a coinciding here pause in the cholinergic signal to disinhibit AC5 from GI. And why is this relevant? So what is the importance of this kind of a result? Well, we said neuromodulators, they seem to represent some quality of the stimulus coming in from cortex. So this really, when you have a result like this, you kind of get the prerequisites about what kind of a stimulus you need in order to enable strengthening of the synapse. So for the D1 emissions, it would mean it needs to be a rewarding stimulus, and whatever else the cholinergic pause represents. So you kind of get, you can kind of get conclusions about the quality of the stimulus that drive learning, actually. And so now we can go into how we got to this result, or what suggested this result. So for that, a little bit of context. So adenylyl cyclases are the enzymes that produce cyclic MP, and cyclic MP is the first second messenger to be discovered in 1951, but despite that, there's still a lot to be studied about these enzymes. So these are the enzymes that from ATP, they produce cyclic MP, they perform this reaction. And we need to go a little bit into the structure of these enzymes because it determines function. So there's 9, in mammals, there's 9 membrane-bound enzymes, and the brain uses all of them. And they're differentially distributed across brain regions, and in the basal ganglia, AC5 is the dominant one. they have these two transmembrane domains. One of the roles is to anchor them in the membrane. At the end, we come to other roles that they have or may have. And then these enzymes also have these two catalytic domains, which need to come together or dimerize in order to form the active site where ATP can bind. And as you all know, catalysis is very sensitive to the precise arrangement of atoms at the active site. And the neat thing about having these two dimers that need to come together and form the active site is that you can just manipulate catalysis just by manipulating their relative positions. And this is indeed how the G proteins regulate AC5. So they each have their own separate binding site in each of the catalytic domains. And when you have these two control points to regulate the enzyme, it is possible to not only do signal transduction from the extracellular side to the intracellular side, but also signal integration of the two incoming signals that activate the two branches. And when it comes to signal integration, it's useful to think about in terms of these logical functions. So depending on what combination of signals is necessary to activate the enzymes, you might have an AND function or an OR function or an XOR function performed by this molecule. And indeed, it turns out that many adenylyl cyclases are in fact AND gate. So they need two coinciding signals to activate the enzyme. And we were wondering what is the case for AC5, since this is the one implicated in reward learning. And what we know qualitatively is summarized in this table. So when no G proteins are active inside the cell, the free form of AC5 has a very low basal rate. We can approximate here with a 0 cyclic MP response. When there's only GI active in the cell, there's AC5 is inhibited, also no cyclic MP response. When only Gαolf is active in the cell, AC5 is stimulated, and this is the case which corresponds to both a peak in dopamine and a pause in acetylcholine. And then for the case when the two G proteins are active, this was not known what happens here. So can the two G proteins bind to AC5 simultaneously? After all, they do have their own separate binding sites on the enzyme. Then if they do really form a ternary complex, such as the one shown here, what is its catalytic activity? Is it active, inactive, or half-active? So we see here that the question of what controls cyclic MP production or what enables the synapses to be strengthened is equivalent to the question of the nature of this possible ternary complex. So this is what we studied here, and we used computational techniques because experiments have not been able to detect this kind of a complex. However, the binary complexes of AC5 with each of the G proteins have been detected and well characterized also in terms of structure, molecular dynamics as well. So it's possible to do comparisons with them. So one thing that we did was Brownian dynamic simulations. So with these, we got estimates of the rates, constants for binding of each of the G proteins to AC5, and also each of the G proteins to the binary complex of AC5 and the other G protein. This was done by our partners in Heidelberg, and it's important to know if these rate constants are fast enough, because the signals in dopamine and acetylcholine are very short, less than half a second. So these need to be fast enough so that otherwise the signals will just pass by, the G protein is deactivated, and they won't get to interact with AC5. And we see a little bit later how we test whether this was enough for binding, whether this was fast enough. And so like this, we see that the ternary complex may form, and then we want to say, okay, it may form, but is it long-lived enough to make any difference? And then what's its catalytic activity if it forms? So for this, our partners at EPFL and Jülich performed molecular dynamic simulations, and they simulated this ternary complex based on results such as, my apologies for the weird looks, the weird borders of this image. Based on values such as how much the backbone of the complex fluctuates, how much hydrogen bonds change in this enzyme, the fluctuations of each amino acid residues, so one of these is shown in this graph, we won't go into it. They were able to determine that this ternary complex is stable indeed. And then also from these molecular dynamics simulations, you can get information about its catalytic activity. So this distance here shown in green between these two helices needs to be large enough for ATP to be able to come in and bind, so the substrate to be able to come in and bind. And this distance is shown here in blue in this graph for the ternary complex, and this you can see is very small, even smaller than the inactive binary complex of AC5 and GI. And this, by comparison to the inactive binary complex, was shown to correspond to a closure of the active site. So in this sense, ATP wouldn't be able to come in and bind here, so it's probably, the ternary complex is probably inactive. You can also get information what happens if ATP somehow managed to come in and bind at the site. For catalysis, it is known that this distance between this oxygen atom and the magnesium cofactor needs to be small enough in order for the cyclization reaction to be performed. And in the course of the molecular dynamics simulation, so ATP inside the ternary complex seems to switch to change conformation so that this distance becomes nearly double to what is needed for catalysis. So this is another implication that this ternary complex is probably inactive. And now we have several pieces of information here that the ternary complex can form its rates of formation, and it's stable and catalytically inactive. We put these in a kinetic model of this signaling module to see what the effects are in terms of cyclic AMP production. Kinetic models, I suppose you all know, are these ordinary differential equation models of bimolecular reactions. We use two versions, one where the ternary complex can form and one where it cannot form in order to see the effect. And these are the results. So here are the modeled peak and pause in the neuromodulators, the associated activation of Gαolf plus the activation of GI. And here are the results of the cyclic AMP normalized to the basal response. In blue are the results is the response when the two neuromodulators signals coincide, one with red with only dopamine and yellow with only acetylcholine. And we see here, when it's not possible to form a ternary complex, you have a response both with coinciding signals and only with a dopamine, only with the dopamine signal. And the case with an inactive ternary complex has a response strongly only in the case of coinciding peak and dip. And this is exactly the way we mentioned in the beginning. In this case, only a dopamine peak will cause the formation of the ternary complex, which is catalytically inactive and keeps this cyclic AMP production, in this case, very low. You need a coinciding disinhibition from the cholinergic by the cholinergic pause, and this is not the case. In this scheme, Gαolf can just come in and bind to AC5 and produce lots of cyclic AMP in the case of only dopamine. And so to summarize, we saw that this question of what controls cyclic AMP production and what enables strengthening of these synapses was equivalent to the question of the nature of this ternary complex. We obtained the rates of informations with Brownian dynamic simulations, and molecular dynamic simulations said it was stable and inactive. When incorporating this in the kinetic model, this suggested that indeed the enzyme responds significantly only in the case of a peak in dopamine and a pause in cholinergic signaling. There's here a set of experiments that I won't go through at the moment. If you have the questions, we can do that. These are explained by an inactive ternary complex. And I can summarize that this was an example of these, how molecular level features gave rise to these functions coming in at the level of a signal transaction cascade and have implications for what triggers or what enables learning. And with adenylyl cyclases are these examples of such computational devices. They are regulated by many different molecules, not only G proteins. So this is kind of just the start maybe of research here. And in terms of these workshops, it's interesting whether also membrane excitations can regulate this enzyme, because the transmembrane domains not only serve to anchor the enzyme into the membrane, but also to properly orient the two catalytic domains, and it has been proposed that they may also regulate catalysis, but no natural ligands have been found for maybe a decade or something. So it will be very interesting to see whether one can have control via membrane pulses over there. And I'll just thank the collaborators and other PhD students, my advisor, and the funders.
Okay, thanks. So, other question?
So, okay, you show there LTP, yeah?
Yeah.
I know what LTP is. What is the link between LTP and cognitive memory now, cognition?
What is the
Why do you think the two things are linked? Where is the link between LTP and the cognition?
Yes, this is. Well, this is a big question in neuroscience, but it is thought that the configuration of synapses at a dendrite enables a neuron to respond to particular patterns. And I need to draw a picture to be able to explain this more. So you have maybe the neuron with the dendrites over there, and then this, let's say, particular pattern of some strong synapses, some weak synapses here, and maybe not too many strong synapses here. This would enable a neuron to respond more strongly via the combination or configurations of synapses there to cortical input coming over there. So somehow it is thought that learning or memory is somehow encoded by one of the things, maybe the strength of the connections, and other aspects of the synapses over there. But this is
This is an assumption, or it is a conclusion. And this is the point. So
no. So I mean, this is I guess this is a hypothesis of a kind of a very vague, let's say, or not
vague link or
not very yeah. So this is a big question in neuroscience. Where do memories lie? This is one, let's say, plausible candidate, but yeah, plausibility arguments. This is
Okay,
this
maybe
is a different question. Yeah.
maybe a short question from Connie, and I think we are pretty late. We should then go to lunch. And before Connie asks a question, I should announce that at 2 o'clock we will have a talk by Rudolf Riegler, and so on. It would be very kind if you were back in time. Yeah. So Connie.
Of course, in Stockholm, the SciLife lab, very big next to Karolinska, has made a transformation to initiatives to computers.
Yes.
A lot, and it's very much debated, and the people who initiated have themselves now turned to be critical about, and you are one of the victims in doing computer simulation.
Yeah.
Now, if we now here want to make relations with the things we are discussing, it would be very important to see whether everything you are talking about is membrane bound. Or are there really, like it looked in the pictures, some of the enzymes and things are bound to a lipid membrane or associated, and others are not considered to be. But, I mean, I would like if everything is bound to membranes. How is it?
So whether everything is bound to the membrane, I don't know, but it seems that almost everything is quite nicely organized, so they don't just float freely around. These Brownian dynamics estimates are for sure underestimates of the true, let's say, rates. And this slide here, so here at least this study was studying the shapes and associations of receptors in the membrane, and at least the receptors for the neuromodulators and the AC5 are bound over there. And in gray underneath, you see the assumed G proteins, which are also there. The G proteins have been shown to be already pre-associated with the catalytic domains of AC5, AC5 to other things inside. So maybe not everything is bound to the membrane, but they seem to be quite well localized and do not just diffuse in the...
So you know of no element which definitely has nothing to do with membranes and is just sitting somewhere in the middle. No, you wouldn't know.
I mean, these anchoring proteins coupled to cytoskeletal proteins lower inside, you have these things which are not so close to the membrane. But this, I would say, for any purposes, this is all pretty much...
We can make a membrane
Yes.
theory
Yeah.
for your simulation.
Okay, maybe we should stop here, we are already late. So thanks again to the speaker. We will have lunch now.